Welcome to episode 2 of module 5 heredity. We're looking at inquiry question 2, how important is it for genetic material to be replicated exactly? Our syllabus reference for this video will model the processes involved in cell replication, including DNA replication using the Watson and Crick model, and construct appropriate representations to model and compare the forms in which DNA exists in eukaryotes and prokaryotes. Our learning intentions will outline the structure and function of DNA, distinguish between DNA and RNA, compare DNA found in prokaryotes and eukaryotes, and describe DNA replication. In 1953, James Watson, Francis Crick, and Maurice Wilkins won the Nobel Prize in Physiology or Medicine for the discovery of DNA. However, it was the contribution of biophysicist Rosalind Franklin that was crucial in puzzling together the structure of DNA. Rosalind, unfortunately, was never officially credited with the discovery, as she died 4 years before the Nobel Prize was awarded. The discovery of the double helix has paved the way for modern molecular biology and greatly impacted fields like genetics, medicine, biotechnology, and forensic science. DNA stands for deoxyribonucleic acid. It is the blueprint of life that functions to store and transmit genetic information of an organism. Some of its prime functions include serving as the set of instructions for the formation of proteins, undergoing replication to form multiple copies, which is essential for cell replicating processes like mitosis and meiosis, and regulating gene expression, which is when certain genes are turned on and off in the development of organisms. DNA is composed of 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. The bases adenine and thymine will always pair together, and guanine and cytosine will always pair together. One way I like to remember is a tree and car in the garage. The bases are like the rungs of a ladder, but to support the rungs, there needs to be a steady backbone. In the case of DNA, the backbone consists of phosphorus and the sugar deoxyribose, hence the name. The complete DNA structure is referred to as a double helix. A nucleotide consists of a base, deoxyribose sugar, and a phosphate. Without getting into too much biochemistry and confusing you all, we need to understand how DNA is read. The double strands of DNA are antiparallel. DNA is read in the 5' to 3' direction. Its antiparallel strand is read in the 3' to 5' direction. This is important when DNA is being copied during DNA replication. 5' to 3' refers to the number on the deoxyribose sugar molecule in which the phosphate attaches. Deoxyribose, the sugar, has a pentacon structure and is a carbon-based molecule. The phosphate binds to the 5th carbon on the sugar, and the sugar binds to the phosphate from the 3rd carbon. This is why it's referred to as 5' to 3'. The antiparallel strand runs from 3' to 5'. DNA and RNA are both nucleic acids but have differences in structure, function, and location in organisms. We know from the previous slides that DNA is a double helix structure composed of 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. RNA, however, is a singular strand, and just like DNA has a backbone consisting of a sugar, in this case ribose, and also phosphate. Thymine is not found in RNA and is replaced by the base uracil. RNA's prime role are involved in gene expression and protein synthesis. We will look more at RNA's involvement in protein synthesis when we look at the formation of proteins in another video. DNA is found in the nucleus of cells, whereas RNA is found throughout the cell in the nucleus, cytoplasm, and ribosomes. If you remember from Year 11 biology, prokaryotes are types of cells that do not contain membrane-bound organelles, such as a nucleus. Organisms such as bacteria and archaea are examples of prokaryotes. Eukaryotes are organisms with membrane-bound organelles and can be unicellular or multicellular, including animals, plants, fungi, and protists. Both cell types contain DNA. However, the structure and the function of DNA is vastly different. In prokaryotes, DNA is single, circular, that floats freely in the cytoplasm, whereas DNA in eukaryotes is linear in shape and located in the nucleus. Chromosomes are tightly packaged structures that carry our genetic information. In eukaryotes, DNA is wound tightly around proteins called histones. Different organisms carry different amounts of chromosomes. For example, humans have 46. In prokaryotes, the DNA is not tightly wound around histones, and there is only one principal chromosome, which may have multiple copies. The replication of DNA is much faster and simpler in prokaryotes than in eukaryotes. This is because eukaryotes have billions more base pairs to contend with. Both prokaryotes and eukaryotes contain extra chromosomal DNA. In prokaryotes, these extra chromosomal DNA are referred to as plasmids. In eukaryotes, there are no plasmids. However, different cell organelles, such as mitochondria and chloroplasts, have their own DNA. DNA replication is the process by which a cell makes an identical copy of its DNA. This process is essential for cell division, growth, and repair. Some key players involved in DNA replication are helicase, an enzyme involved in binding to DNA and unzipping it into two strands, creating a replication fork; DNA polymerase III, an enzyme that catalyzes the formation of new DNA strands using a pre-existing template of DNA; primase, an enzyme involved in initiating the start point for DNA polymerase to begin making new strands; and ligase, an enzyme involved in ligating or joining DNA fragments together. DNA replication begins at specific sites on the DNA molecule called origins. Enzymes called DNA helicases unwind and separate the two strands of the double helix by breaking the bonds in between the bases. This creates a replication fork. One of the separated DNA strands is known as the leading strand. The enzyme primase makes primers to instruct where DNA polymerase begins to add complementary bases. DNA polymerase builds the new strand of DNA in the 5' to 3' end. The other strand is known as the lagging strand. It has this name because it lags behind. This is because it attempts to keep up with the leading strand, but cannot, as DNA polymerase adds the bases in the 3' to 5' direction. RNA primers have to continuously move back and forth along the lagging strand to instruct DNA polymerase to add bases. Because DNA polymerase struggles to keep up with the leading strand, shorter fragments of DNA are built. These are called Okazaki fragments. In between these Okazaki fragments are gaps where DNA polymerase has not added bases, again because it cannot keep up with the leading strand. The enzyme ligase fixes this by gluing the fragments together so there are no gaps. At the end of DNA replication, you have two strands of DNA that are identical. This process is called semi-conservative because each new strand contains a copy of the original strand and a new strand. The process of DNA replication is important in processes like mitosis and meiosis, and we'll model this process in class to help get a better understanding. And that is the end of episode 2. Thank you for watching.